Today's video is on physiology and homeostasis. So this is a physiology class. What physiology is studying the biological function, aka how the body works. And so we're interested in looking at whether our cells are functioning normally. We want to look at specific mechanisms. We're going to learn different mechanisms throughout the body, and whether one thing happens, some sort of cause, what sort of effect it'll have. You took a human anatomy class prior to this class, and so you are familiar with the anatomical terms, and now we're going to learn about how everything works. So just to introduce some parts, our body has intracellular compartments as well as extracellular compartments. Intracellular means inside the cell, and we have fluid in there, and so this is called the intracellular fluid, which is inside our cells. And then on the outside of our cells, we have our extracellular fluid. But our extracellular fluid is actually made out of two different components. We have fluid that is found in our blood, which is our blood plasma. And then we have our actual extracellular fluid that is outside of our cells, which we also call interstitial fluid. So there's two components there. And, the way that we consider how our body is functioning, we have to talk about homeostasis. And so maybe you're aware of this idea, maybe you learned about it in anatomy, but homeostasis is when there are factors in our human body that are maintained under a stable environment. So it's, relatively stable. I should write relatively stable here. And I'm left-handed, and I hate how my left hand is moving the slides. So what that means is it is not constantly constant. For example, you probably know what your relatively stable body temperature is. In Fahrenheit, it's 98 degrees Fahrenheit. But in science, we use Celsius, so it's about 37 degrees Celsius. That does not mean that your body is always 37 degrees Celsius. What that means is that we try to stay within a range of 37 degrees. And if you go outside of that range, if you have a fever, that tells us that there might be a disease or something that is impacting our homeostasis, and something might be wrong. And so looking at how our body is acting and whether it's maintained under homeostasis is really important. So body temperature is one example, but you can think of heart rate. Maybe you can consider, or maybe your own resting heart rate, blood pressure, oxygen levels. I'm just giving some examples here. Glucose levels in the blood, which we'll talk about in just a second, and body temperature, of course. And we'll talk about a couple other, pH is a good one. We'll talk about other factors, but there are a lot of factors that are maintained within homeostasis. And again, that does not mean it is constant, but it stays within a range. And if we go outside of that range, either too low or too high, that could possibly ring, an alarm in our body and tell us that something might be wrong. In human anatomy, you learned about your body systems, which comprise of different organs, and we will learn this semester how our body systems work together to maintain homeostasis. Because maintaining homeostasis is essential for our cells' survival. Perhaps you can think of some examples of cells that you learned that are working under specific conditions. Let me give you an example of that. Your stomach cells. So maintaining a specific homeostasis for specific cells is going to be important for their functionality. We know that our cells make up our tissues, which then make up our organs, which then make our organ systems. So the majority of the time, homeostasis is maintained by negative feedback. And negative feedback is called negative because it's going to do the opposite of what the stimulus is. And we're going to talk about a few examples here. So this is one of the main ways that our body maintains homeostasis, and it's a major way that our functionality changes when we have different stimuli occurring. So some examples of this, we've been talking about body temperature, and glucose in the blood. We're going to talk about these a little more deeply. And heart rate. We're going to talk about this later on this semester. The way that our body uses negative feedback is by these negative feedback loops. They have these specific pathways, and each pathway has each of these three parts. We have sensors in our pathway that are going to sense change or sense a stimulus. These also might be known as receptors, or and sensory receptors. We're going to have an integrating center. Oftentimes this could be the brain or the spinal cord, which are going to process that information, and they're going to interpret it and send instructions to a third party, which is an effector. An effector is going to cause or going to affect some change. So this could be muscles, this could be a gland, or a visceral organ, but there's going to be a change that is going to impact the stimulus that our receptors first sensed. In negative feedback loops, the effector usually counters the change that was caused by the stimulus. So let me give you an example here, and we'll continue our example of body temperature. So let's continue to use body temperature as an example. Say there were sensors in our skin, for example, that sensed that our body temperature was high. Maybe on a hot day, or maybe after you went for a run. Those sensors would send information to our integrating system, which, oftentimes is our brain or spinal cord. I could just write CNS. And it's going to cause some sort of effect. And if we're talking about body temperature here, it's going to do the opposite of that, right? To try and decrease our body temperature. And it might do that by activating our sweat glands. That is one way in which we regulate our body temperature. And so you can see here that the effector is doing the opposite of what the sensor received, the stimulus that the sensor received. And this is why it's called negative feedback. It's doing the opposite. And so it's kind of like a seesaw. If you know what a seesaw is, it's like where kids can go up and down, and you're doing the opposite of what the other child is. On the opposite side here, say our body temperature was too low. We're going to sense that somehow. The integrating center, the central nervous system, is going to process that information and then also send information to an effector, which is going to affect some change. So if our body temperature was low, one way that our body temperature could increase is by constricting blood vessels so that, your fingers don't get as much blood because we lose a lot of heat in our extremities. Another way is by shivering. And in this way, we can do the opposite of what we sensed, what was the first stimulus, and increase our body temperature again. So again, this is negative feedback. Oftentimes in homeostasis, there are opposing effectors that are going to change conditions in opposite directions. And so I just gave a great example of body temperature and how we sweat when our body temperature is too high. or we shiver when our body temperature is too low. And so these are considered antagonistic effectors because they do the opposite to change the stimulus in opposite directions. So again, as we describe what homeostasis is, there is never an absolute constant, but there is actually more of a movement of constancy within this range. And so we call that dynamic constancy, which is kind of like an oxymoron, right? It's changing within those two, within this range. So negative feedback, 99% of the time is how we maintain homeostasis. There is the rare instance of positive feedback. And I will give you some examples of this, but positive feedback is when you have a stimulus and your body responds by amplifying that stimulus and increasing it and increasing it. And this may seem wild and crazy because this is not actually pushing our body back towards homeostasis. It's not pushing our body back towards that main, dynamic constancy within that range, right? It's kind of pushing it outside of that range. But this positive feedback is often usually part of an overall negative feedback. And let me give you an example of that. Say you cut yourself and you're bleeding, I should draw this in red. You're bleeding. The way we actually produce blood clots using platelets is by positive feedback. Your platelets surround each other and they attract each other even more and more and more and more and more, amplifying this change until it blocks our ruptured blood vessel, closing it, and then bringing the body back to negative feedback. So that's a great example of how it's part of an overall action towards negative feedback. Other examples of this are female related. So if you think about ovulation, ovulation is triggered by a surge of luteinizing hormone, and this goes up, up, up until you release an egg, which then brings you back towards your homeostasis via negative feedback. Another example is uterus contractions and oxytocin secretion during labor. If you've ever had a baby before, those contractions increase and increase and increase and increase until you have ejection of your baby. And then that brings you back to homeostasis. There are two ways in which we regulate homeostasis. Intrinsically, intrinsic sounds like inside. It means the change comes from inside. And so cells within the organ can sense the change and express the effect themselves. An example of this, your heart. Your heart can change its own heart rate. Extrinsic regulation is the use of an external organ, kind of like your brain, right? It's an external organ outside of the receptors that are going to affect some change. You should remember this from human anatomy, hopefully. Our nervous system innervates our organs via our, neurons, which we'll talk about more. I'm just drawing some right here with these nerve fibers. Or our endocrine system releases hormone, which are kind of like chemical messengers that are going to impact target organs only if they have the receptors for those hormones.